in terms of biophysical tools of looking at entire animals, organisms, humans, tissues in humans. And this is starting to get at the techniques that, are they biophysics techniques, or are they more biology, even ecology, more shared and core facilities in the School of Medicine that you'll see, but you know, biophysics can span quite a scale, and I definitely think these fall under those areas, but you'll see them more often used, yeah, in those other types of departments than I think you would see in a physics department, technically. So when I'm talking about the tissue and organism level techniques, I mean, this is a deviation from, like, we've mostly been talking about molecules for most of the semester. We then moved on to cells. And the nice thing about molecules and cells is that you can nicely put them in a little test tube, or you've seen in many of the techniques, like in BioBox, like a 96 well plate. But if you have like a whole, let's say a mouse, I don't know if I can draw a mouse. That looks like a cat. But if you have a whole organism, you can't just throw them or an animal into a test tube or a 96 well plate or something like that. So that's why there's different techniques that are used in a different suite of methods that are applied at that scales. I'd say that's like due to size, due to optical transparency, since a whole bunch of the other methods we were talking about use light and electromagnetic radiation, or I'd say like due to sensitivity or damage to the sample with like high magnetic fields, if you're talking about NMR or things like that. So that's, I guess, the motivation of this lecture or this topic. But let's first start out with like, what model organisms are there or are used that are greater than the size of the cell? Since we talked about many of the cellular model organisms in a past lecture. So this is where we're going to start with the breakout groups. I think this should be pretty, hopefully a straightforward breakout group of each of you guys. I'd like you in your group. I've arranged it from smallest organism to largest, and from fruit fly, C. elegans, zebrafish to mice, and why, identifying why that organism is used as a model organism. If you guys can find a cool video or paper, like I've listed that these are like news aggregators for scientific news that like universities put out, that I'm guessing that these have plenty of results and options for these model organisms, if you can find something interesting that they've been used for. And then if there's a facility on Case's campus that manages this model, I know for some of these, there are a shared core facility or that takes care of some of these organisms. So individual labs don't have to do that. Some of these are easier to work with. So if you can't find a core facility, can you find a faculty member doing research with this organism? So if you just type in CWRU and that title, can you find something like that? So are there any questions with the breakout groups? Okay, so let me, if group one, if you guys have someone who is a spokesperson, go ahead and share. Oh, maybe not everybody was there.
Forgot to designate a spokesperson, but we each have a couple different things. So I found a paper about, I am group one, right? Fruit flies. Okay. Anyways, so I found a paper about sleep homeostasis in fruit flies. It's a 2020 paper. And the first thing they wanted to do is they wanted to see if fruit flies would make a good model organism to assess the mechanisms and functions of sleep in mammals. And so they did that by essentially point for point comparing if the mechanisms of sleep in fruit flies were similar to the mechanisms in sleep in mammals. And the two main things they were assessing was this one thing called sleep rebound, which is after you've been sleep deprived for a very long period of time, and then you finally get a chance to sleep, you enter REM sleep faster, and your REM, your rapid eye movement, is deeper and it's quicker. And they found that sleep rebound does occur in fruit flies just like it does in mammals, and the degree of sleep rebound is dependent on how sleep deprived you were prior to entering REM. And then they also tested if sleep deprivation impaired motor function and vigilance. And it was kind of cool how they did that. They essentially put flies in a maze. They let the flies get used to the maze, and then they made half of the maze very, very hot. And they assessed how long it took for the fruit flies to escape the heat, which is very mean. But they found that in flies that had been sleep deprived, it took them longer to move to the cooler sections of the maze. And then they also found that sleep homeostasis, so the desire to sleep after you've been awake for long periods of time, was conserved between males and females, just like it is in humans. So they concluded that fruit flies are a good model organism for investigating sleep in mammals.
How did they keep the fruit flies awake?
Actually, I don't know. That's a good question. Yeah.
Do they blare loud music? Do they keep the lights on? I wonder.
I was going to say, I saw something about laser beams in there, but I'm not sure if that was actually involved.
Okay. Cool. And anyone else in group one, did you guys identify any general characteristics of why they're used for a model or benefits, or if there's anyone on campus doing fruit fly work?
So the general gist of why they're used is because they're low cost and they reproduce really quickly, so rapid generation. And they are a simple model that is close to humans. What's the thing they mentioned, that they have close to the same number of genes, I think it was. And actually have a video I could share that I found might be fun to look at. I don't know if that's fine.
Yeah, that's fine.
But
it's enabled if it works. Okay.
Oh, and there's an image. Well, that we want to share. Let me share the video.
Sent a link as well of fruit flies in space, I guess.
Yeah.
They're actually open.
I don't know if the audio is working. Essentially, they were looking at fruit flies fighting each other to moral behavior that you would see in humans. And they were looking at things like depression and addiction. And the way they get them to fight each other is by shining laser light at them. Well, no, just, I think just red light was.
So they don't like the laser light, I guess.
No, it wasn't a laser, it was, I don't know, it just had red light they found it.
Yep, pretty cool.
But yeah, but this would trigger neurotransmitters that made them aggressive.
Mm-hmm.
Yeah, that light.
Okay. Cool. And did you guys find anyone on campus doing fruit fly research, or did you not look? It's fine if you didn't.
No, but quick
yeah,
sidebar. Oh, sorry.
Oh yeah, you go ahead.
Apparently they kept the fruit flies awake by putting them in a box, and then whenever they slept, they just dropped the box, and that would wake them up.
Okay. Rough life for those fruit flies. I know Nicole Crown in biology. She's newer faculty, and her lab is all fruit fly stuff. And with quarantine, she's been having to go in just to have. She's had a lot of genetic mutations or forms of fruit flies, and she has to go in and make sure like you can't just kill those fruit fly lines off. So that was a big thing during pretty much everything was shut down, that she had to go in and keep those guys living and going on. So with some of the genetic mutations that we talked about, I guess in the last lecture, yeah. And Andrew just shared another group in biology. So I think when I was in high school or undergrad, we did like looking at the eye colors of different fruit flies and stuff. So I don't know if any of you guys have worked with fruit flies, even just like in intro courses. That is a testament to how cheap they are and easy to use. Even high schoolers can do that. Okay. Cool. Should we move on to group 2? I think C. elegans was the next one.
So similar to Drosophila, C. elegans is another small animal model that is very cheap, has really high fecundity. They produce like 1,000 eggs per day. So you can do a number of studies. They have a really defined developmental pathway, however, so that makes them really efficient for studying any form of development, as well as inducing mutations that affect development, because you know exactly what cells are affected in what order. So you can like identify phenotypic changes very, very simply. Because they produce so fast, have such a short life cycle, inducing these mutations throughout can lend to systems biology approaches where you're inducing tons of mutations and identifying thousands of potential drug targets within the model. Let me share my screen for a video. Okay. So we just picked unlike Drosophila, where you can like have really neat behavior, moving the flies around and like attracting them to various substances. C. elegans videos are a little bit more boring. I'd say we couldn't really find that many cool videos. So we just showed a quick video of the development. I'll skip around, but this is an embryo of C. elegans. And so people have these like high resolution cameras focused on the embryo, and they can watch it develop and see all these cellular events happening. Let me speed it forward. So it quickly becomes a multicellular organism. The worm stays transparent throughout the lifespan. So you can view cellular changes even as it's fully developed, which is really neat. So it'll split, and then now it's like having a defined shape, turning into a worm in the egg.
Yeah, probably used a lot in developmental biology of the cell differentiation.
Yeah, a ton. So eventually it splits and starts like, I don't know, slithering around.
Yeah.
And there are no C. elegans facilities. I'd say they're used much less than Drosophila in general, but we found Michael Crowder, who's like a physiology MD PhD. Like I said here, he does these unbiased screening approaches. So he'll have a ton of C. elegans models, and he'll induce point mutations or just mutations throughout the C. elegans genome at various locations. And then he'll do a screen of like all the phenotypic changes and identify like a phenotype of interest. So like if he wants to see, he finds C. elegans that are like super long for some reason. So he like identifies he induces mutations throughout the genome. He finds 50 such C. elegans that are super long, and he looks at what genes are mutants. So now like he identifies genes that are correlated to changing C. elegans size. Like that's an example of one of these systems biology screening studies that can be heavily applied in this model. That is all for us.
Cool. And also I know C. elegans are used quite a bit in neurology focused studies as well with neurons. So when we talked about like the electrophysiology methods of like the voltage probe and stuff, I've seen that used a lot in C. elegans, and you actually see an increase in even scientists and electrical engineering departments when they're expanding into more of the medical and bio type research. That's where I've seen the first, I guess, biophysics type research show up in electrical engineering departments, is usually looking at neural signaling pathways and C. elegans. So it even spans over there in the College of Engineering. So cool. So let's move on to group 3, I think was the zebrafish.
Right, okay, so the zebrafish is this small tropical or Asian fish. It grows very quickly. It's pretty easy to maintain. It's a vertebrate, so I guess that it has like a lot of the same major organs compared to humans. and it's apparently a bit easier to take care of than, say, rats or mice, cheaper to maintain, certainly. And other than that, they've been used since, I think, the 60s as a model organism. And they actually have even though they're fish, they share 70% of their genes with humans. And actually, in terms of human diseases, 84% of genes in zebrafish match the disease genes in humans. Let's see what else. The genome has also been fully sequenced. And yeah, so it's mainly for looking at diseases, which we'll show a video in a second that kind of discusses some research into human disease using these zebrafish. Other than that, before that, let me share my screen real quick. We found at Case Western, we did find a core facility in the School of Medicine that raises zebrafish for research, though some of the links on this website are broken, and it seems like this page hasn't been updated for several years, so I don't know if this facility is still open, but
that's a pretty common theme with core facilities at Case. And I'll tell you, at the university level, they're trying to work on that. So
okay, well, at least at one time there has been, in the recent past 5 or 6 years, there was a zebrafish facility in the School of Medicine here at Case. And then I think if one of my partners can share that video.
So here's the video. I'll play the video. It's 1 minute and 46 seconds, and it's going to talk about 3 human diseases that zebrafish have helped treat. So here we go.
FBR, we want to share with you 3 diseases that the zebrafish has helped researchers study and find treatments for. The zebrafish is a tropical fish that is native to Southeast Asia that is widely and commonly used in biomedical research for multiple reasons, such as their low cost, their transparent design, which allows for easy observation during testing, and its genetic structure that shares 70% of the same genes as humans. Number 1, heart diseases. Because of their ability to regenerate cells, scientists have been able to study the reasoning for various defects in a human heart shape, size, and function. For example, zebrafish have been the ideal model in studies on how to repair cardiac scars that were formed from heart attacks. Number 2, deafness. Zebrafish have been used in studies of age-related hearing loss, which has been proven to affect nearly half of Americans over 75. Through the study of zebrafish, researchers were able to learn more about the inner ear of humans and various reasons for the cause of deafness. Number 3 is cancer. Zebrafish have also proven to be a great model to study different forms of cancer. For example, zebrafish have allowed scientists to find safer and less toxic methods for treating pediatric cancer. These are just 3 diseases that the zebrafish have helped researchers study and find treatments for. They have continuously proven to be a great ally in the efforts of biomedical research to help improve the lives of humankind. For more information, please feel free to visit us at FB Research.
Cool, thanks for sharing. And yeah, that optical transparency that they touched on, I mean, that was why, I think it was in problem set 2. There was that example of looking at protein folding within zebrafish embryos. So you can do very high resolution imaging due to their transparency. And then I liked how they pointed out the deafness, because in that same group, which I worked in a little bit during my postdoc, there was even someone doing like tracking of fish after they like scared them with sound. So again, just like dropping the fruit flies, like lots of not fun tests for some of these animals. Okay, let's move on to the last group, the common, I guess, mouse studies.
Okay. Yeah, so on campus, there are a decent number of facilities that have mice or look at mice, and also a lot of labs just have their own separate mice facilities. So it seems like there's not one dedicated core, but there's like the case transgenic and targeting facility, and there's also the mouse behavioral phenotyping core. I guess there are just a lot of aspects that people are interested in. They're mammals, so they're just fairly like genetically similar and behaviorally similar to humans, but they have like a much shorter life cycle, so you can get multiple generations a lot more quickly. Within a set sample or within a set sample size or population, you can make them very genetically homogeneous, like excluding like third sex, just by inbreeding them a lot. Their behavioral like mechanisms are just fairly well documented, so it's easy to phenotype them. They're also just very cheap. And for our graphic, we have this cool mouse brain slice here. But they actually imaged this like through the skull using 3-photon instead of like thinning the skull or just taking a brain slice, which we thought was pretty cool. Yeah.
So was it like, do you know if it was like infrared light then?
Yeah. So they were using IR photons. And 3-photon is just 2 photons, but with 3 photons, as we learned. Yeah.
Cool. So any other questions, comments on, I guess, what people found in with the different organisms? Okay, cool. So let me get back to sharing my screen. Give me 1 second. Sorry, I forgot a slide. Okay, sorry, thanks for your patience. These are just some summaries, and I think you guys touched on a lot of these with, I guess, my pen, of course, stopped working. You guys touch on quite a few of these things for each of the organisms of the short life cycle, the cost, transparency, the sequence similarity. I didn't know it was like 1,000 eggs a day for C. elegans. That's pretty impressive and the like. I also just want to mention if you're working with one of these organisms, that you have to go through the Institutional Review Board or the IRB. You'll hear that terminology. So even if you are like keeping these fruit flies awake for a long time or scaring the zebrafish or heating them up, you have to ensure that you're doing it in an ethical way. And that's part of the Institutional Review Board. So that's common with any work with organisms and even cells or recombinant DNA that the NIH requires this, also like CASE requires this, that's a board of, I believe, faculty members that will then review what your plans and your procedures are and ensure that they're matching up with what is accepted in the field. I guess an interesting aside with the IRB is, I know like in the past lab that I worked in that worked with zebrafish, with those embryos, they weren't allowed to just like discard them after they did the studies because they had to be discarded in a very specific way. So they did just have a freezer that was just like literally just filled with, it was like the zebrafish graveyard, and they had to wait until like a certain time where they could do a certain procedure to get rid of those. So that was just something I thought was interesting with that. And I'm very confident that the zebrafish core facility on campus still exists, but you probably have to reach out to the person in charge of it, and they just haven't maintained their website. Okay. Cool. So also with these model organisms, I wanted to touch on some methods that I've seen people do with the organisms. And I think like Julia's example of putting animals through mazes, I've seen in terms of tracking, that this is getting into the area of biomechanics and motor dynamics within an organism. We've talked about like single molecule motors and the like, or forces, but how the mechanics that allow organisms to move and function and respond quickly. This can be done tracking quite slowly with conventional cameras, but I've seen, especially with insect studies, doing ultra-fast tracking. So to see every single thing that's going on. And this camera I've shown here, that's through a company called Phantom that I've seen most commonly used. And their cameras can go 6,000 frames per second, or they can even get up to like around 50,000 frames per second if you're using like a subsection of the camera chip. So this example here, it has a scientific CMOS detector. And instead of like when we were talking about fluorescence, a lot of the emphasis in that camera technology is on detecting every single photon and focusing on quantum yield. Here, these detectors are focused on the speed. But if you want to get these very fancy, nice cameras, they're going to be running closer to like, they're going to be in the upper tens of thousands of dollars. So maybe like 80K or something around there. And depending if you're getting top of the line or not, even pushing up to 100K. So those are some of the fastest cameras on the market that I've seen. But you can also do tracking at much slower speeds. So down here is an example that I've seen quite a bit in mouse studies. So mice hate being wet, and you'll have, in this example here, this is a pool of water, and there's one pedestal that's in that pool. And you'll introduce the mice to the pool and they'll swim around until they find this pedestal, and then they can dry off and stand on it. And typically the water is obscured, or the pedestal color is obscured, so they can't see it, that they have to search around. So you can see here that they have a camera overhead and they track where the mouse goes. So this is a wild type mouse here that is able to find it quite quickly. They do the swim repeatedly so then they know where that pedestal is. This is a protein with a mouse with a mutation, so it has tau aggregates that occur in Alzheimer's disease. So you can see it has to take a lot of time to find where that pedestal is. And then this is with I believe, a treatment to turn the gene off. And you can see it recovers that fast ability to find where that pedestal is, and it can dry off. So then it's a test at the mouse level, the organism level, of seeing what the effect is of a genetic treatment to then relate to the animal behavior. Cool. So any questions at all in terms about like the organisms or animal type models and studies, insect studies? So a few other methods, techniques that at the tissue organism level that I want to touch on include histology, which is microscopy of tissues and understanding like the micro anatomy. So you'll commonly see this in terms of pathology and diagnosing diseases. Is where I'll see it more in the medical field used more commonly, but it's also used in biophysical studies that, yeah, once you introduce mutations, certain treatments, do you see a change in the distribution of the anatomy of different types of tissues, especially like if you're trying to identify cancer, mutated cells versus healthy cells, this type of imaging can help. There's 4 different types of tissues. in general classes that you'll see epithelium. So that's all the cell types that would be at the edges of organs, like your skin is an example of epithelium cells. There's connective tissue, nervous tissue, and muscle tissue. And then you can see in these images that there's also subclasses of each of those tissues. So histology is trying to identify within a slice of tissue what is the organization and what types of tissues are present. I would classify this as quite a specialized technique. And the fact that being able to collect these images and identify the cells requires training. And it's required to, I guess, you have to fix the tissue. Next thing you have to do is section that. And you can imagine if you fix, let's say like the mouse brain that Sean showed us, if you fix that mouse brain and then you have to section it and slice it, that has to be done very carefully so you're not exerting a bunch of force, disrupting that tissue, introducing artifacts there. So that's quite specialized as well. After you section it, you then have to stain the tissue. So then there's colors and contrast so you can actually identify what's going on. And then of course, identification. So there's quite a bit of labor involved in this process. And then even looking at the resulting images. So you can see down here, these are sectioned, fixed, stained tissue types. Then looking under the microscope, you get images that look like this. You can see it's micron scales here, tens of microns. But getting these images and then being able to identify which tissue types it is takes some training. And I'll say when I read papers where it's histology, and this one is nicely labeled, but many times the histology images will just have like an arrow pointing to stuff. And I don't have histology training. For my untrained eye, it's very hard for me to tell the difference. So I appreciate the people who are specialists in this area. So because it is specialized, there's typically cores that take care of this. So at Case, in the School of Medicine, the histology core is in the Cleveland Digestive Diseases Research Core. So I'm guessing a lot of their staining has to do with digestion, but I'm guessing they also have histology services for other types of diseases and tissue types as well. Okay. Any questions with histology, or have any of you guys had experience with it?
Yeah, I do a lot of histology in my lab.
Do you feel like, am I correct in saying that it's a pretty specialized technique? I at least feel like it is. I don't know if that's how the school of medicine people.
It depends on what model you use, because I'm using a mouse. So when I sacrifice a mouse, like mice are worth a lot. And so when I sacrifice it, I try and get as much, like, I try to make it worth it when I'm sacrificing an animal, because they are worth a lot of money. And so I collect a number of organs: spleen, kidney, liver, bone marrow, peripheral blood, and I perform histology on all of them.
Mm-hmm. Yeah, that's a lot of organs and tissue and stuff. And Julia, do you have any comments? I also see that you put in the chat that you've worked with histology.
Oh, no comments. I definitely don't have as much experience as Jack.
Okay. Cool. Yeah. But yeah, this histology is getting at, I guess, yeah, more towards medicine. And also the last things I want to touch on are methods that I would say are more, are they biophysics? Yes, but also I would say, what's the line between medical physics and biophysics? And Rose, am I correct in remembering that you have a degree in medical physics as well?
No, that's correct. I do.
Okay. So if I say anything wrong here, please correct me. But do you have a clear definition of medical physics versus biophysics? Medical physics dealing with patients, or I don't know if you want to provide your definition.
So medical physics really is working on imaging scanners, like techniques also. And mainly we do magnetic resonance imaging, but we also work on ionizing radiation, like CT radiographic units. And the idea is to minimize dose to the patient and at the same time getting like the best image. And that it's a mix of quality control, technology, and protocols too, in all different modalities. So yeah, and mainly these stuff are used in radiology and diagnostic. So that's it shortly.
Yeah, very clear. And Rose, you're the first person that I've met that's started in medical physics and then moved to, I guess, a PhD in physics, but I guess your research is still in the medical physics area. That I've known several people who have done physics research, like literally like very fundamental physics research, and then have gone afterwards and gotten their degree in medical physics following like their PhD in physics. So they're some of the most trained people I know. And I think it's very cool to see how physics STEM applies to treating and helping people out and the like. So I know Professor Gao has had a PhD student go and get a medical physics degree. And then one of my friends from my PhD is at LSU now in Louisiana and does medical physics. And I know a lot of his work is, there's the work of helping doctors figure out like decreasing the amount of radiation to treat patients with these different techniques. But I know also a big component of his work is managing the instrumentation as well, and helping the vendors with the calibrations or ensuring that the imaging equipment is working correctly. So I feel like that has a lot of overlap with, like, he was building like microscopes and working on optical tables, but that's a common thing I think with experimental physics of making sure your equipment is working correctly as well. So I do just want to touch briefly, I guess, on MRI and then some of the other imaging techniques like Rose mentioned, like CT and the like, so you guys are aware of them. But you can take entire, like I mentioned, entire degrees, entire courses in medical physics, and especially MRI here on campus in the physics department, you can take 2 courses on that. MRI is based on the physical principles that we talked about with NMR. So can you guys throw in the chat or just unmute, like, what region of the electromagnetic spectrum were we using in NMR and like what phenomenon? If you guys recall. Any comments? So we were with radio waves in the electromagnetic spectrum and taking advantage of nuclear spin in the presence of magnetic fields. So with MRI, obviously has to have a different type of setup compared to an NMR. Like, we can't throw a mouse or a human in a little glass NMR tube and the like. So you have to have the electromagnetic coil has a relatively large diameter, maybe something around 70 centimeters or so, and the magnetic field ranges are in the 1 to 7 Tesla range. So not as crazy strong as when you guys were exploring the magnetic facility down in Florida and the like. And remembering when we talked about NMR, that the emission from the nuclear spins are going to be very sensitive to the local environment. So that's going to give you the information about what's surrounding the given tissues there. So something unique with MRI as opposed to NMR is that you want to get your images in 3 dimensions. So you're going to have a special arrangement of how that coil moves and how the patient moves relative to one another. So since you're producing 3D images, those are then called in your images voxels instead of pixels. That just means it's a 3-dimensional space. And the resolutions in MRI are typically around tens of microns. But depending on if you're using some advanced technology or not, you can push those limits. And then MRI, I think as Rose mentioned, it can be used for diagnostics. So you're trying to identify like the heterogeneities of the variety of signals that you're getting in tissues. But there's also functional MRI as well. So fMRI is what you'll hear that referred to. And that's where you want to monitor changes in dynamics. And many times this is applied to metabolism. And to monitor those dynamics and changes over time, typically these are done in the presence of a contrast agent, and there's quite a few contrast agents that could be introduced. I'm going to focus here on this right-hand side of using gadolinium. So that element is often used as a contrast agent, where when you're looking at studies where they're developing contrast agents, you'll see this is a very common type of image that you'll see in MRI agent development, where this is, I believe it's in a 96 well plate where they introduce the different contrast agents. I think here is with the different concentrations, and just putting that in MRI, collecting the image and seeing what the signal to background is. So the bottom row here is your control of the water, and these are different forms of gadolinium in different carbon nanomaterials. So you can see here some carbon chains, a buckyball over here that has the gadolinium inside. And then that can be introduced to your mouse model. Maybe you'll have some targeting agent that the contrast agent will go to a specific site, and then you can collect images where here you're seeing over time in a mouse. I think this might be the spleen, I can't remember specifically, spleen or liver, maybe the kidney, now that I'm looking at more detail, of being able to identify the different features with the different contrast agents. So this is an example. So I believe this guy over here, this is commercial versus like the nanoscientists developing their gadolinium carbon nanotubes shown there. Some other methods that I would classify under biomedical physics would be like X-ray techniques, CAT or CT scans, where this stands for computerized tomography. So this is using X-ray imaging with a scanning geometry similar to MRI. So you can get information, both 2D, but more importantly, 3D images of the X-ray attenuation. So this gives you nice levels of detection. It's typically used for some cancer diagnostics, and it can identify tumors down to around like 2 centimeters with, I would say, conventional methods, but of course, always the cutting edge might have better resolutions. There's also, in addition to detecting X-rays, there's also detection of photons and gamma rays. So single photon CT is using radionucleotide labels. And those will target and bind to specific molecules in the body, and it will emit gamma rays. So the radionucleotide labels that you would introduce, these would be iodine 111, 123, or 131. The most common one is technetium, which I need my periodic table, 99, which in the leak book they talk about. This is so commonly used that there was a shortage, I think in the 90s, and they highlight that story, that then they had to develop molybdenum. 99 to have a new radionucleotide label that could work similarly to the technetium. That was actually like a problem in the imaging community. A similar gamma ray detection would be positron emission tomography, or PET. So this is using a positron emission instead of direct gamma emission. So positrons are the antimatter equivalent of electrons, and they'll interact very quickly with an electron nearby and then cause gamma ray emission. So this uses different radionuclides. So this would be like carbon 11, nitrogen 13, oxygen 15, fluorine 18. So you can see these are more common, that those will emit the positron and then combine with electron to get gamma ray emission. And then finally, using last method I want to mention is ultrasound, where you're using sound waves and you're measuring the acoustic impedance. And there's many variations and specialties in ultrasound. There's Doppler ultrasound, where you're also not just looking at the impedance, you're looking at the wavelength change due to the Doppler effect as that travels through tissues. So these are imaging methods applied at the animal or human level in medical biophysics that you might see come up. And especially over in biomedical engineering, you'll also see these methods show up as well. And so any questions there, or Rose, any critiques? It was supposed to be very brief and I guess somewhat broad. Cool. And then one very last thing, because I realized I didn't talk about this at all during the class, and I feel like I should mention it, that antibodies are used with some molecular techniques that I wanted to make sure that I mentioned. ELISA, which would be enzyme linked immunoabsorbent assay. Where this is taking advantage of antibodies and their high affinity for a specific antigen. So antibodies are created naturally by our immune system to target something very specific, like the spike protein on the COVID virus. And these techniques, ELISA takes advantage of that. There's different arrangements of how antibodies can interact, but you would have in your 96 well plate, you would have a capture antibody fixed to that surface. Then you would introduce your solution, and if it has your target protein present, that would then bind. Then you do another flow of another antibody that would then detect that protein and would have a fluorescent marker or colorimetric marker. So there's different sandwiching ways that and arrangements of how many antibodies and how many proteins and how many labels, but these are typically done in screening methods in the 96 well plate, where each one you introduce a different solution and the color will tell you if it's there. A Western blot is taking advantage of ELISA at the end, but you're including gel electrophoresis at the start, and then you would do ELISA, or instead of ELISA with like a colorimetric marker, you could also do like staining and then do like photographic development of that even. So you'll see in many biophysical papers, ELISA or Western blot images. Western blot images are typically not as clean as this, that you'll have band broadening and smearing and the like. And I would say that's one of the most commonly manipulated images that I've seen people get in trouble for changing in publications that you shouldn't change that much.